أعوذ بالله من الشيطان اللعين الرجيم بسم الله الرحمن الرحيم اللهم صل على محمد وآل محمد وعجل فرجهم. Friends, today we are going to discuss non-structural protein ELISA test. This is actually a kit which is used to detect non-structural proteins of the virus. Non-structural proteins of the virus are mainly present in FMD virus. In FMD virus, there are two main types of proteins which are present. Number one, structural proteins, and number two, non-structural proteins. Structural proteins and non-structural proteins. When an animal is infected with the virus, which is a field strain, then that virus contains both structural proteins and non-structural proteins. So in that animal, antibodies which are developed, they are against both structural proteins and non-structural proteins. But in vaccines, for vaccine preparation purpose, we remove non-structural proteins of the virus, FMD virus. Non-structural protein portion of FMD virus is removed for the use of for the purpose of the vaccine formation. So when we prepare vaccine, we remove non-structural protein portion of the FMD virus. And when we vaccinate the animal with that virus, from which the non-structural proteins have been removed, then in that animal, the antibodies which are developed, which are raised after 1 to 12 days, they will be against only and only structural proteins. So this ELISA kit is based upon this principle, that if this ELISA kit is basically used to differentiate between between the animals which are infected with the field strain and the animals which are vaccinated with the strain from which non-structural proteins have been removed. So results may be like this: if animal is infected with the virus and also the animal is vaccinated, then the ELISA non-structural protein ELISA test will be positive, will show positive result, because the virus from the field contains both structural and non-structural proteins, and this ELISA kit will detect non-structural protein portion present in the field virus, and result will be positive. Result will be positive on kit. It means that non-structural proteins are present, meaning that animal has suffered from the field strain as well, recently or somewhere in the past. Similarly, if animal is only infected with the field virus, which contains both the structural and non-structural proteins, but animal is not vaccinated, unvaccinated, and we take the serum sample from that animal, the result will again be positive on non-structural protein ELISA kit, because the virus from the field contains the both structural proteins as well as non-structural proteins, and those non-structural protein portion will be detected on the ELISA kit and test will be positive. And if animal is vaccinated with that strain from which the non-structural protein portion of the FMD virus has been removed from vaccinal. For the preparation of vaccines, we remove the non-structural protein portion of the virus and then we use it. And if animal is only vaccinated and not has suffered from the field strain somewhere in the past or recently, then if we take the blood sample and separate serum from that blood sample and put it on the non-structural protein ELISA kit, test will be negative because there there is no availability of non-structural protein in the vaccinal strain. And when there is no availability of non-structural protein in the vaccinal strain, no antibodies has developed in that animal against those non-structural proteins, so they will not be detected. And similarly, if animal is not vaccinated and also not infected with the field strain, then again on non-structural protein ELISA kit test will be negative, because in vaccinal strain again we have not vaccinated the animal with the vaccinal strain, which contains only structural protein portion, and animal is also not infected with the field strain, which will contain both structural proteins and non-structural proteins. Both are not present, so blood of the animal will not contain the non-structural protein portion, and again also not contain the structural protein portion, so result on non-structural protein ELISA kit will be negative. So this is basically like sandwich ELISA test. In sandwich ELISA test, what is the basic principle? That we have 96 wells polystyrene ELISA kit having captured antibodies coated in the base. We take the blood sample from the animal and separate the serum from that blood sample. Basically 10 ml blood should be taken, some should be taken as a sample, and then we separate the serum from that blood sample, and that serum is added. That serum might contain the antibodies, and those antibodies might be both against non-structural protein portion and structural protein portions. And if the serum contains non-structural protein antibodies, then that antibodies attach those those antibodies will attach to the captured antibodies, and when attachment will occur, they will attach, and we will incubate the plate for 16 hours. After 16 hours washing will be done, and those antibodies which are against non-structural protein portions, they are attached with captured antibodies. They will not be removed, and then we will add secondary antibodies which are enzyme linked as well. So we will add at this step we will add enzyme linked secondary antibodies. These are also again very specific to the attached antibodies. And if enzyme linked, we will incubate the plate again for 1 hour and at 37 degrees centigrade, and after 1 hour we will washing step will be done again, and after washing, these secondary enzyme linked antibodies will not be removed because attachment has been occurred properly, because non-structural proteins captured antibodies are there, then specific non-structural proteins antibodies were present in the serum. They have been attached with captured antibodies, and then after washing they have not been removed, and now secondary enzyme linked antibodies are again will be attached with these antibodies. After that, we will check the result by the addition of the substrate. After the addition of enzyme linked secondary antibodies, and when we add the substrate, we will wait for 20 minutes again for the development of the color. And if enzyme linked secondary antibodies were present, were attached with the uh, anti non-structural protein antibodies, then color will be shown, color will be developed, and test will be regarded as positive. It means that non-structural proteins are present, and the presence and the detection of non-structural proteins will indicate that animal has suffered from the viral strain, field strain, and animal has suffered recently from the field strain. The positive test means animal has suffered, has been infected with the field strain as well, whether it is vaccinated or not. And if test is negative, it means that animal has not suffered from the field strain, from the field virus, and it is only and only the vaccinal strain which is which has developed the antibodies against structural proteins. So thank you very much. Jazakallahu khaira. Allahumma salli ala Muhammadin wa ali Muhammad wa ajil farajahum.